Paul Nurse won the Nobel Prize for Medicine in 2001. In this series of 5 lectures, Paul talks about how the great ideas of biology underpin our modern understanding of the living world.
Now, on the whole, us biologists tend not to talk too much about great ideas and grand theories. We leave those usually to the physicists. We biologists tend to be more comfortable with details, with particulars. What we like to do is to count things and list things. If I was a 19th century biologist, I would count the numbers of hairs on a beetle's leg. If I was an ecologist, I would count and list the species in a habitat. If I was a molecular biologist, which in fact I am, I would sequence genes and determine the proteins and RNAs that are in a cell. So we tend to describe things, but perhaps we don't focus enough on the great ideas of biology. But biology does have great ideas, and this evening I'm going to talk about 4 of those ideas, 4 which in my opinion are very important, and I'm going to point in the direction of a 5th, which is yet to be fully formulated. My second idea, the gene as the basis of heredity. Now, from ancient times, it was observed that particular characteristics were passed down from generation to generation, from parents to offspring. And so it was clear that people, that scientists, that others, that philosophers wondered what was the basis of this heredity. And this was a problem that was being tackled indirectly in the middle of the 19th century by botanists interested in improving crops. They were crossing plants with different characteristics and trying to breed plants which had better characteristics. And what they observed was that often characters seemed to blend, sometimes characters seemed to disappear with one cross, sometimes those characters reappeared when a subsequent cross was carried out. So it was confusing. This problem was solved by the great, indeed the first, geneticist, the gardening monk Gregor Mendel. He himself was trained as a physicist. He carried out, and indeed was encouraged to carry out, receiving, as we would now know, research grants to carry out his research work. He built a very large greenhouse and had a garden. He was, however, tempted to become an administrator later in his life and became the abbot and stopped doing science. But before he did that, he carried out some wonderful experiments in plant genetics. He argued that to sort out what was happening in these crosses, with all these characters disappearing and reappearing, it was very important to accurately catalogue the numbers and types of plant forms and the characters that were being generated, and describe how they appeared in the different generations of plant crosses. And there were 3 reasons, I would say, for his success in solving this problem. The first reason was he chose material that he could understand. He did trial crosses with a variety of different plant species until he found a species that seemed to make sense. You could say he was cheating. He decided only to study what he could understand. That's really important. Biology is complex, and we have to use material that helps us through the complexity. If you start by understanding something simpler, it's easier to move on to something complex. He worked on the pea, and he chose the pea firstly because there were clearly defined different strains with different characteristics, tall plants, short plants, different coloured seeds, different coloured flowers, so that you could cross different plants and follow very clear characteristics. He also, as I've already indicated, recorded very accurately what phenotypes, that is what the qualities of these plants were, and counted them. Biologists tend not to be so quantitative. They are more now, but remember he was trained as a physicist, he was an amateur meteorologist, and he counted what he saw. And then also, after that, the third reason I think for his success was he generated a theory or a law to explain his observations and then went on to test that theory and law. Again, a feature that we associate more with the physicists than with biologists. Now, what he observed is that in the first generation of the crosses he was making with the pea, that often one character in one of the two parents dominated in the progeny, but that the other character re-emerged in the second generation, and often re-emerged in distinct ratios. And you may remember from your school studies, because you often you should have been exposed to this at school, that the very famous 3-to-1 ratio of these different characters emerged in the second generation. What you probably don't know, because I only found out a few months ago, was that the same 3-to-1 ratio was described by Charles Darwin working with snapdragons in 1868 at the same time as Mendel. But what Charles Darwin did was simply recorded what he saw and did not try to make a theory or try to make sense of that in the same way as Mendel went on to do. Now, what he went on to do was essentially to propose a particulate theory of heredity. And what I mean by particulate theory is that he proposed that genes acted as particles which determined the characteristics of plants. Implicit in that statement, incidentally, is that those genes, those particles, conveyed information. And I shall return to information towards the end of my talk. He also proposed that in any particular plant, there were two particles that were important for determining a particular characteristic. But when that plant reproduced and made gametes, only one of those particles was passed on into each gamete. On fertilization, those two particles, one from one parent, one from another, reformed the double act, the two particles from the pollen from the egg in the offspring. And he also proposed that one particle might dominate over the other in determining what the character was. Now, I can't explain all the logic of that, but I hope you got a sense of the type of thinking that Mendel carried out. And truthfully, it was a brilliant piece of abstract reasoning, simply based on some of the ideas here. You see a yellow seeded plant crossed to a green one. You see the yellow one dominating in the first generation. You see the green re-emerging again in the second generation. With these observations and a brilliant piece of abstract reasoning, he proposed a particulate theory for heredity. The only problem was nobody took any notice of Gregor Mendel. It was a very clever paper. It was read by others. I've come across it in the Encyclopedia Britannica of the 1880s, so it wasn't simply ignored, but nobody took any notice of his theories. Until 35 years later, when, again, one of these rather extraordinary coincidences which are so common in science, 3 independent scientific groups carrying out plant breeding experiments repeated the same type of experiments, came up with the same observations and the same theory. One of those groups tried to sneak it into publication without acknowledging Gregor Mendel. One of the others, who was publishing a bit behind, pointed the error out to the first one. In the end, they all had to acknowledge the gardening monk, who had scooped them 35 years before. Now, why were these observations and these theories taken seriously in 1900 to 1901 and not in 1865? I don't really know the answer to that, but I will give you an idea. And that is that during that period, chromosomes were discovered. Chromosomes, of course, are the structures in cells which are made up of genes. This is a 19th century picture of an onion tip, and you can see the threads appearing as cells divide. And seeing the threads appear as cells divide, knowing that the cell, as I've already explained, is the basic unit of life, of course made biologists think that maybe the chromosomes might be the structures that were responsible for heredity. And this was clinched with respect to Mendel with an observation made by van Beneden looking at a nematode worm. And once again, this illustrates the importance of using the appropriate material. The nematode worm, or the species he was looking at, because if I remember my zoology correctly, there's over 50,000 species of nematode worm. This particular one has 4 chromosomes, so you could count them very easily. And what van Beneden observed was that when this worm reproduced and made gametes, then only 2 of those chromosomes appeared in each gamete. So you had the reductional division that Mendel had postulated when only half of the determinants, what we would now call genes, were passed on into the gametes. So here we see that the abstract reasoning of Mendel was being supported by the concrete observation of biology. And I think it's quite often that biologists like seeing abstract reasoning confirmed by concrete observation. And this we see with the van Beneden chromosomes here. Now, this idea of heredity was turned in a wonderful series of experiments in the 20th century into molecular genetics. And this started with the question, what are genes made of? And this was discovered by Avery, MacLeod and McCarty at the Rockefeller University, of which I'm now president, in 1944. And what they discovered was that if they extracted from one form of a pneumococcus bacteria DNA, deoxyribonucleic acid, and then sprinkled it onto another form of pneumococcus, then you would see at a certain frequency a transformation from the first strain into the second strain. And that suggested that it was DNA, deoxyribonucleic acid, that was causing this transformation from what were called smooth colonies to rough colonies. This was not actually well accepted at the time. It took some years to be accepted. And I remember being taught about this in the late 1960s when I was an undergraduate. So imagine my surprise when I went to Rockefeller University in 2003. And this little old man came up to me to introduce himself at a cocktail party. And he said his name was Mac McCarty. And I said something fairly stupid, of course, which was, "You're not the Mac McCarty." To which he said, "Yes, I am the Mac McCarty." And here we had McCarty, who published this great paper in 1944, still coming to the university in his 90s in 2003. I have to say, I thought he was dead already in 1968. Having discovered that DNA was the molecule of heredity, of course, then the story that I'm sure is much more familiar to you was of Watson and Crick and Rosalind Franklin and Wilkins, all contributing to the determination of the structure of DNA and the double helix that you see here. And this was then extended further, particularly by Francis Crick, into what has become to know as the central dogma of molecular biology, where the DNA encodes the RNA and is transcribed into RNA, which follows the sequence of the DNA nucleotides, and then the sequence of RNA determines the sequence of amino acids in the proteins that are encoded by that RNA. And that means that the information encoded in the DNA is eventually translated into a sequence of amino acids that make up the protein that determines so much of what goes on in a cell. So we understand where the information is stored and how that information is translated into the characteristics of the living organism. So this is my second great idea, that the gene is the basis of heredity, that it can explain the genotype to phenotype, how a genotype can give rise to the phenotype, and it continues to be of great importance. It has tremendous implications for what we are, how much are what we are determined by our genes that we inherit from our parents? How much of what we are is determined by our environmental influences? And these are very important problems and also important for us understanding what we are. If you go into any bookshop and you look at the popular science section, you would think this is probably the only idea in science.